Right, this Friday we have the exam, but I also said we should keep the Monday. We should keep the Monday after the spring break open because there is an opportunity for some extra credit. Let's see it first. Okay, so right, this guy here will be at CSU Monday after spring break, and there's a lecture at 11 in the morning, and it's in the Law Students Theater. I think the lecture is very relevant for this class, and I'd like you to go there. And I'll give you some extra credit. And the way that will work is I will ask you a question, which I will post on the internet a week or so after. And then you can turn that in together with exam corrections at that time, and you can just get a couple of extra points. Now, my default is 20, but I like excitement. So, if I shoot in the funny head there, and I take 5 points off, all right? But I get 3 shots because I have the right to practice. Or actually, if you like 3 shots and make it twice, you get only 10 points. Okay? That's not in. And I'll make it from here, right? That was close. One more. Well, that was harder actually. Okay, so 20 points. I knew this explanation. Hey, what you should do is you can Google this. And I'll make an announcement. I'll make an announcement on the internet, so you know where, when, etc. But you just have to go, and I'll ask you a question afterwards. I'm not telling you in advance. Okay, that's that. Then the order of business today, we have to finish some stuff. And there we go. Some people have asked me to put the answers for the task 3 and 4 on the internet. I've done that for task 3. For 4, there's still, of course, the deadline hasn't passed, so that's too early. But I've put some scribbled answers which give you some reasoning for the questions on task 3. I've put that on the website. It's in the folder tasks. It's called the scribbles or something like that. And for task 4, the same will be following. All right. So, let's finish the topic of microfilaments and how they are relevant for us. Microfilaments are made up of actin. This is a part of the cytoskeleton. In most cells, this part of the cytoskeleton is very dynamic. We can have several functions of actin. It helps shape the cell. It also helps cell locomotion, crawling action. By actually the polymerization of actin filaments, a cell gets its surface pushed forward. Now that mechanism involves a couple of other components, including membrane receptors that we will see later. But also important for actin is that it can work together with what we call motor proteins, which can walk over the actin filaments and take some cargo along. Here we have a diagram of such a motor. It's a complex motor. and it takes a vesicle which it moves over an actin filament. So this is one mechanism by which organelles in the cell can be placed in specific positions. So the motors that do this walking are called myosin. They're always called myosin if they walk over actin. And there are various kinds of myosin, and the various kinds have a Roman numeral name, like 1, 2, 5. All of them move to the plus end of an actin filament. And all of them have a structure as depicted here, which is composed essentially of a cargo connecting tail and two head parts that bind ATP that are responsible for the bulking. So we always have a motor head region. In this myosin we have two of those motor heads. And here we have the tail. That tail can connect to some kind of a cargo. The cargo can actually be another myosin. So then you can have two myosins that are tail-tail connected, and it can walk over actin filaments, which results in the contraction of that actin cytoskeleton. We'll see that later. Things are really strange things happen. So how do we know that myosin is a motor and that it uses ATP? Well, we can do some simple tests. Myosin can be purified and it can be bound to a glass plate. And many proteins actually bind spontaneously to glass. That is not so hard. There are a lot of hydrophilic groups. Glass tends to be quite sticky to proteins. So myosin can stick to that. A myosin will stick either with its head or with its tail. If it sticks with its head, it's dead. For this assay, we don't care about it. But if it sticks with its tail, then it looks like this. This is a myosin, and the tail sticking out. Now, actin can be purified, and it can be polymerized in vitro. And if we fluorescently label those actin filaments, we can format. So the actin filaments can be allowed to bind to the myosin, and then we can give ATP and see what happens. So we can see that ATP-dependent walking just under a microscope, a fluorescent microscope. So here we have an actin filament, and let's just imagine this is a picture, fluorescence picture, shot from this glass plate here. There's an actin filament, and a couple seconds later, you see the actin filament has moved, according to this arrow. You see all these filaments here moving. Now, based on this type of assay, you can test and see what you really need. So we know we need ATP, and the ATP must be in the form that it can be hydrolyzed. There are other forms of ATP in which one of the oxygens that is on the terminal phosphate is replaced with a sulfur, and that's called ATP gamma S, and this cannot be hydrolyzed. And if we add this, we see that actually the actin filaments can be released from the myosin, but they no longer move in a specific direction. So using analogs of ATP or by flooding the system with ADP, we can make observations, and this leads to the following model. The binding of ATP to myosin actually causes it to release from the actin. Then the hydrolysis of ATP by the myosin causes the myosin to rebind the actin, typically at another position. Then when inorganic phosphate is released from the myosin, a power stroke happens in which the myosin actually shifts the actin over its surface, or the myosin moves relative to the actin. So we know there's a full cycle of ATP hydrolysis, release of PI, rebinding of ATP, after release of ADP. And this gives the walking motion of myosin. All right, now, to give you some relief, we will come back to the action of motor proteins much more after the spring break. For now, what's very interesting and important to finish is the way in which muscle contraction works and in which it is regulated, because it comes back to the action potential and the neuromuscular junction. So a little bit of anatomy. If we have a muscle here, then actually in those muscles there are very long cells, and those cells, the muscle cells, are cells that have many nuclei. These are actually fused cells. All cells are of course formed by cell division, but for skeletal muscle cells, after the divisions are complete, there's fusion of cells, so you get cells with many nuclei. Syncytia, they're called. So a syncytium is a cell with many nuclei. Then in those cells, we have these very long fibers, and if we look at the ultrastructure, we find that there's ultimately a contractile unit, which we call the sarcomer. And the business part of the sarcomer is drawn here. And this sarcomer is composed of actin filaments, so the microfilaments in the yellowish orange, and the myosin in the pink. And you see that the myosin is tail to tail connected. Here this is the head parts of these rounded parts, like the golf club ends. There are the heads, and the tails here are all connected. So there's a bundle of myosin, and there are actin filaments. This is the contractile unit. And if we do a cross-section through the contractile units, and we look at it in the EM, we see here the myosin, that's the darkest spots, the thick filaments, as it's called, and the microfilaments, the thin filaments. Right here in the middle, we only have the tail elements of the thick filaments. So you need to remember actin and myosin in this sarcomer contractile unit. And the way muscle contraction works is what happens, the myosin heads walk over the actin filaments, and they walk in the plus end direction, and the plus end is here at these Z discs. So, upon walking of the myosin filaments, these actin filaments are pulled in together. And you can see this in an EM, and you can even see it in a light microscope picture. In an EM picture, we see here uncontracted muscle and contracted muscle. This is the sarcomer, this is the sarcomer. What it takes for this contraction to happen is that there's ATP and there's calcium in high concentration in the cell. That means millimolar concentrations of calcium. So the ATP is required because myosin requires ATP. Right. The calcium is required because calcium is a trigger in this process. Okay. So now we need to understand how calcium triggers the process. And to understand that, we must acknowledge that there are a couple of proteins missing from our picture here. There are a couple of proteins missing. The important protein is tropomyosin. Tropomyosin is a very long protein, and in this very nice picture, which is an EM picture, sort of a, based on shadowing image, we have here a microfilament, so actin filament, and covered in this case is a long protein called tropomyosin. And this picture here without calcium shows that the tropomyosin is in close contact with the actin filament. Now it turns out that if calcium is added, the tropomyosin is slightly shifted in position, and this now exposes the binding sites for the myosin on the actin filaments. So these red sites here are potential binding sites for myosin, which are exposed when calcium is present. So tropomyosin shifts position, which allows the myosin binding sites to become exposed. But tropomyosin itself doesn't bind to calcium. That's another protein that is closely associated, and that's troponin. Troponin is the calcium sensor. Calcium is often used as a signaling molecule. We've seen it before. It was used as a signaling molecule for the release of neurotransmitter. Remember that? Here it is also used as a signaling molecule. Calcium can bind to glutamate and aspartate amino acids in specific proteins and affect the conformation of those calcium-responsive proteins. And troponin is one of them. When troponin binds calcium, it shifts conformation, and that conformational shift leads to the slight push of tropomyosin, so that actin now can bind myosin. So here we have a diagram of how this might look. Troponin in green, the calcium sensor. When calcium binds, it pushes a little bit on the tropomyosin, and the tropomyosin shifts position. And that exposes the binding sites for the myosin motorheads. So in words, the increasing calcium in the cytosol causes this slight change in the shape of troponin that pushes the tropomyosin away, so the myosin binding sites on the actin filaments are exposed, and then when there is ATP, and of course there is, when there is ATP, myosin will walk over actin and the sarcomer will contract, and then the entire muscle cell contracts and leads to the contraction of the entire muscle. So that's how it works. So we have the myosin filaments, they're there. We have the actin filaments, they're there. But tropomyosin binds to actin and it prevents myosin binding by default, unless troponin, upon calcium binding, pushes tropomyosin away. There are subunits of troponin. I'm not so interested in knowing those. But you should remember these four players. All right. So how do we get the calcium to be the trigger? Well, let's look at some more anatomy. Here we have our motor neuron, and it ends up here on a muscle cell. That muscle cell is relatively large. There's a neuromuscular junction. We have a synapse there, remember? There's acetylcholine release. There is depolarization of this muscle cell membrane because there are ligand-gated sodium channels. They bind to acetylcholine and they open. Sodium flows in. We get some depolarization locally of the membrane, the muscle cell. Then voltage-gated channels in that muscle cell membrane open, and the entire muscle cell membrane depolarizes. That muscle cell membrane has strange tubes that it forms that run deep into the cell and allow for close contact with another membrane system, which is called the sarcoplasmic reticulum. And we can think of this as an ER-like structure in the muscle cell. It's a very extended tube-like structure. And this sarcoplasmic reticulum is a reservoir for calcium. There are gated channels in the sarcoplasmic reticulum, voltage-gated channels that respond to the depolarization of the plasma membrane in the transverse tubules. So when the plasma membrane depolarizes, when there's an action potential there, the sarcoplasmic reticulum calcium channels open, and then the calcium which is stored in this sarcoplasmic reticulum, or SR or SER, goes out into the cytoplasm. So that calcium is then released. That release causes the contraction because the calcium, which is normally at very low concentration in cells, is suddenly at millimolar concentrations, and it triggers that response by troponin, which triggers the conformation shift in tropomyosin, which allows myosin to walk. Now, how do you relax? To relax, you need to get the calcium back into the SER, and that's done by means of a P-class pump, which uses ATP. This is actually the best studied pump, one of the best studies, if not the best studied membrane protein. This calcium pump is very efficient. It uses ATP, moves it back into the cytoplasmic reticulum. So when calcium is out of the cytosol, there is masking now of the myosin binding sites, and the muscle, the sarcomer, can relax because the contact between myosin and actin is broken. Because tropomyosin now blocks that again. Now you ask the question, yeah, but it was walking over it, the myosin. So how can the tropomyosin actually get back? Well, in the presence of ATP, myosin lets go. So as long as there's ATP in the cell, you actually can get relaxation. This is why, probably, if animals have died, they become rather stiff, because their ATP concentrations go down, which affects their muscles in two ways. They don't get the calcium back into the cytoplasmic reticulum because that takes ATP, and they don't get the myosin to release from the actin. Okay, so the sequence of events that you should know, and you should know to combine with what we know already from the neuromuscular junction, is an action potential arrived at the motor neuron. There were some presynaptic events there, including calcium release in that neuron, which led to release of neurotransmitter by vesicle fusion. That neurotransmitter is acetylcholine. It's released in the synapse. It diffuses over the synapse. It reaches the postsynaptic membrane. That's the muscle cell membrane in this case. That leads to the opening of sodium channels, ligand-gated sodium channels. That leads to a local depolarization. That leads to further depolarization of the target membrane, the muscle cell membrane, because there's also voltage-gated channels there. When the complete muscle membrane depolarizes, that depolarization reaches the transverse tubules, and then via a close contact, calcium channels in the cytoplasmic reticulum open, calcium moves into the cytosol, down gradient. That binds with troponin. That leads to a conformational change in troponin, which pushes tropomyosin away. Actin now can bind myosin. Myosin walks over actin. The sarcomer shortens. As long as there's ATP. For relaxation, you need calcium to go back. In the presence of ATP, myosin will let go of the actin. And we know that the voltage-gated channels have a refractory period. After a short period that they're open, they close again, and resting channels in the plasma membrane let the muscle cell return to the original resting potential, holding the relaxation. Now this is something you should know, because it adds to the events in the neuromuscular junction. Good. All right. So I have made the exam for Friday. I have something to say. I definitely think I did my utmost best to make this a fair exam, a doable exam, it's straightforward. I think I took a lot of questions from the tasks, I really did. And of course, some of the numbers may have been changed. So you need to know what to do. You cannot just learn the answer. You need to know what to do. That's one. But I think the exam is straightforward and doable. I did my utmost best to take any trickiness out. I really did. Yet, I know it's challenging, and I know this exam will be a difference maker. It will be. Last Monday, I spoke with the instructor of the class afterwards, and it seems there's no class here on Friday. afterwards. Not 100% sure. But if this turns out to be correct, I'll give you an extra 15 minutes. Which I think you won't really have to need. I think you can do this exam in about 30-35 minutes. But that's me. So, but I think it's going to be doable. I don't want anybody to be more nervous than you need to be. Okay. Now, what was this stuff all about? What was this last 3 weeks all about? Maybe you can use this projector here. Oh, we have this one. Okay. All right. Our very important topic was electrochemical gradients, remember? And energy in biology. And I've gotten some questions by various students. And that has given me the idea that it's really just review a few basic things. So, what makes a reaction go in biology? The reaction is possible, if I'm writing out of your field of vision, okay? If this here, the free energy change, is negative. So delta G is the free energy change. And what does that mean, free energy? Free energy means energy available to do work in the system. And we can define the change in free energy as the Gibbs free energy after minus the Gibbs free energy before. So we know, and most students don't have a problem remembering this, that there tends to be a loss in free energy for reactions that we observe. There's less energy in the system after something has happened. So if we just take a number, yeah? And if, for instance, before the reaction we had 10 kilocalorie per mole units of energy in the system, and if after that was less, so maybe it was 7, then what do we have for the delta G? We have 7 minus 10, that's negative 3. So you see that if the free energy after the reaction is less than what we had before, then the change in the free energy is negative. That is a spontaneous reaction, that's a reaction that is possible. Every reaction that we observe has a negative delta G. Now if we have a system which is composed of here a membrane, and we have some ions on both sides of the membrane, or just some molecules, then we can ask the question, what is the free energy change for the move of those molecules over the membrane? So let's first go with a simple example, and let's just say that we're looking at an uncharged molecule such as glucose. And let's say we have a pathway for that glucose, some kind of a syn-porter, or let's say a uniporter, okay? That glucose can move through. And we know that this glucose will move always from a higher to a lower concentration. Nobody has a problem with that, right? So let's just say we have 100 millimolar here, and we have 10 millimolar of glucose on the other side, on the right side. And the glucose will move in that direction. So how big is the change in free energy that we get for this reaction? We can calculate that change in free energy. It follows from some basic equations of physical chemistry that you actually do not have to know. If you're ever caring about it, to know it at an exam, I will give you equations on the back page. You do not really need them. But let's go that complicated way. And physical chemistry just tells us that the change in free energy. I will move it up. Thank you. There's a very small screen here that I can see myself. And this doesn't work. So the change in free energy in response to this, for this molecule moving in response to this concentration equals, it's a negative 1.4 kilocalorie, left hammer, it's bad for you, per mole times, I'm going to go to the next line here, the log of the concentration at the point where we start over the concentration at the point where we intend to move. CO concentration at the point of origin over concentration where we intend to move. It's 100 over 10. 100 over 10 is 10. The log of 10 is 1. So we have negative 1.4 kilocalorie per mole for this reaction. I'm telling you, you do not really need to remember that formula. There's another and easier way in which we could go. Because this formula actually tells us something. It tells us really that the free energy of 1.4 kilocalorie per mole equals the free energy stored in a concentration ratio of 10 to the first.
Did you move it up here, please?
Yeah, I got you there. And then you can call this a package of energy, and I like that. Because I think it simplifies things. So whenever